Well, welcome everybody. So thanks so much for carving out some time today. I am not Brad Power. I'm pinch hitting for him. He's at a conference. So I'm Brian McCloskey. Rick Stanton is in Yosemite right now, taking advantage of the beauty that provides. But today we're fortunate to have Sheeno. Sheeno, can you pronounce your last name?
It's Thyparambil.
Okay.
Thyparambil, but I go by Sheeno.
Okay, I'm gonna keep it at Sheeno. Sheeno's going to talk about mass spectrometry based clinical proteomics for oncology in regulated environments. Sheeno is the Senior Director of R&D of MProbe Precision Oncology Division. He has extensive experience in developing, deploying clinical diagnostics products. He is the co-inventor on 29 US issued patents related to the use of mass spec for the development of clinical assays. His specialization is in integration of molecular oncology, multi-omic analysis, and biomarker data for drug development programs. He's the site head for MProbe Rockville unit, which is a CLIA certified CAP accredited lab where he manages a team of scientists, medical doctors, and clinical and R&D staff that is responsible for delivering the clinical report to the oncologist for informed decision-making. And I am particularly excited about this discussion because, as some of you know, I just had a biopsy about a month ago. I have 5 cores. I've used one of them for whole exome sequencing, but I have 4 more at my disposal to shed light on my disease. And so I'm hopeful that I can have some breakthroughs and be able to leverage Sheeno's technology. So I will turn the baton over to Sheeno. And thank you so much, Sheeno, and welcome.
Thank you. Thank you for the introduction. Let me see if I can share the screen. There I am. I'm just going to share everything. Go from there. Can you see the screen?
Yes.
Yes.
All right. Screen share mode. Okay. So,
Sheeno, can you maybe go into presentation mode? We're seeing your slide presenter mode.
How would I do that?
I think if you go to display settings, I think you can find it up there.
Is that better?
Bingo.
Okay. All right. So.
And then, sorry, one more thing, Sheeno. I think there's like a little presenter mode.
Oh, so it's so you can hear
down on the right hand side, I think. Looks like maybe you're in it. Just below that, your cursor, I think. Okay, maybe that's it. We can see it.
Okay. So, so today I'll be talking about the clinical guidance from proteomics reports that we have had, and we have had the opportunity to work with thousands of patient samples. And so, what are the lessons that we have learned in different cancer types, and then perhaps a few prostate samples that we have run over the years. So I get asked this question a lot. That's why I just wanted to put this thing up front. So who's MProbe? What have we done, and how did we come into this precision oncology space? We started, I should say, this MProbe is my new overlord. I'm just going to get the presenter mode from here. It's my new overlord, but the company really started in 2001 as expression pathology, and this was a scientist by the name Dave Chrisman, who'd stepped out from the National Cancer Institute and developed this technology of extracting peptides from formalin-fixed paraffin-embedded blocks in tissue. So our patent got issued in 2008, and then I joined in 2009. And then since 2012, we became a CLIA certified lab, a CAP accredited lab, and we've been running clinical samples since 2012. got acquired in 2015 by Nantomics, and we were able to integrate both genomics and proteomics for patients and doctors. And then finally in 2019, mProbe, which is based in San Francisco, was able to get the proteomics lab from the Nantomics. So what does this all mean? I know over the years, we have run possibly 10,000 clinical samples. I want to say half of them are actually the CLIA report that went out. The other was really for R&D, mostly for pharma, but we definitely have a large collection of mass spec-based tissue proteomics data. So I'm preaching to the choir here, but the DNA to RNA to protein to metabolite, and we focus, at least at mProbe, we focus on the protein side of things and also on the metabolite side of things. I'll focus today on my talk on the protein side of things. So I'll just dive deep. I'll just go directly into our workflow. So this is how it all happens. So we have a test requisition form that is signed off by the oncologist. Our client services team will work with the PAT department to get that block. More often than not, we don't get that block, especially if it is with university systems. So we end up sending our kit to them, and our kit basically contains this slide here that's known as the director slides. This is a laser microdissection compatible slides. It's your regular glass slide that's coated with some proprietary coating on it, so that you can stick it into a laser, and then a pathologist comes in there and only marks off the tumor areas. So, and you can this happens with a prostate sample, and they just mark off only the tumor areas, and you can get directly, without no addition of anything, no coating, nothing, on the tumor sample into a tube, just the formalin fixed paraffin embedded tumor material. So once it is in this format, the key invention that happened about a decade plus ago was how to extract from this fixed tissue peptides that you could shoot straight into the mass spec. And from there, if we shoot them straight in the mass spec, there's no additional steps other than just spinning them down and adding certain buffers in there. And then it'll go from there into the mass spec. The beauty about mass spectrometry is the fact that mass spectrometry has been used for a lot of testing in the past, right? For if you're thinking about your vitamin D test that you would get from your physician's office, that's mass spec based test. Thinking about drugs of abuse test, that's a mass spec based test. So this has actually been in the clinical world, just that we haven't known much about it. What we essentially did was, marrying this side of the world with the mass spectrometry side of things. So mass spectrometry really comes in two flavors, if you will. So one is the discovery proteomics, where you're asking this question, hey, what's in my tube? I do not know what's in my tube. That's discovery proteomics. What we are doing here is basically targeted proteomics, where we're asking this question, how much of a protein of interest, let's say HER2, is there in this tube? So in our particular case, we are looking for 72 biomarkers in a single go that we are looking for, quantitating the levels of these biomarkers. And then 5 days later, there is a clinical report that is issued. So from start to finish, this is a 5-day process. It takes two slides. So you're really looking at, so it's a 10 micron thickness. So you're really looking at, 2 to 3 slides, 20 to 30 micron thickness of slides. And then we ask for an H&E slide. Sometimes folks won't send that. So fine, just give us an H&E image and we'll go from there. So this
Sheeno, sorry. This is a pretty detailed slide. I think you are going to need to go in presenter mode. If you there's a icon down at the lower right, right next to the little slide. Yeah, that one. I think that'll do it.
Yeah, let me do one thing. I think I'm getting into. I'm projecting into my screen. I'm just going to take it off, and we'll just go into a presenter mode from here.
There you go. Much better. Much better. Yeah. Thank you.
So perfect. Now,
it's a great slide, so I didn't want to miss it.
No, I connected it to my external monitor, and so that always messes things up. So I just said,
Okay,
all
we're
right,
good. We're good.
take it. So this is a process from start to finish. This is what kind of happens behind the scenes. A lot of our time is honestly spent sometimes here wrangling the tissue out from the pathology labs, and so that's not a pretty process. But once it is in-house, 5 days later, there's a clinical report that goes to the oncologist, and then we end up connecting with the oncologist and saying, hey, this is what we saw, so that they can get more depth of what came in our clinical report. Just more mass spec speak, but essentially we are adding heavy peptides, and then we can quantitate exactly the levels of 72 biomarkers in that sample. So I'll just skip some of these things. The one thing I just wanted to point out is that the bone mets. So this is a process that is actually compatible when you have bone in the tissue. So when you have bone in the tissue, what people do is they actually take an acid and then decalcify it. So when you take acid decalcification protocol, you end up destroying the DNA, or you end up destroying the shape of the protein. So this makes it not compatible for genomic testing or immunohistochemistry testing. So mass spectrometry, we don't have that problem because we're really looking at the linear sequence of the protein. Okay, perfect. That's all technical speak. I just want to put ourselves in the shoes of an oncologist and then say, hey, if a patient is presenting to me, what do I have? What options? So stepping back, really, I can either go with the standard of care options, right? There is a lot of standard of care guidelines out there and those options. Then the other options, which is something that we've seen oncologists deploy, especially in the metastatic or late stage setting, is they will go into off-label use. And it's actually not uncommon to, if you look at the previous JCO reports about 10 years ago, they said 1/3 of all drugs that is given in oncology is actually from off-label use, and that's not indicated on FDA prescribing information. So there's plenty off-label use that's going on. The other option is I can put this patient into a clinical trial. And how do I decide? That's basically what's in my diagnostic report that comes out. So these are the three major ways that an oncologist would treat a patient. So the 72 biomarkers that we have, not all of them are listed here, but broadly speaking, these biomarkers are either meant for chemotherapy, or they are meant for targeted therapy or for clinical trials. So the chemotherapy angle is something that a lot of oncologists are interested in this particular test, because there are a lot of tests out there that will tell you, they'll probably inform you more on the targeted therapy side of things, but less so on the chemotherapy side of things. What type of chemo agent should I give? There are tests that will tell you whether you need a chemo or not, but you will not, those tests won't necessarily dwell into what type of chemotherapy I should give. So you probably recognize some of the most common ones here, especially for prostate cancer. You're thinking about the taxane-based regimens, paclitaxel, docetaxel. This is Abraxane, which is an taxane that is coated with albumin. And then there's a lot of these markers here. I mean, those markers here are indicating whether you should or should not receive platinum-based therapy. The much more newer side of things, if you will, the newer, because this is one of the targets that has been used most recently in the antibody-drug conjugates. I'll talk about antibody-drug conjugates, but long story short, the payload on that is basically going to attack this particular molecule, topoisomerase 1, and this is an old drug. And essentially, how do you deliver potent chemotherapy into a patient's tumor? So, so some of these biomarkers that we have is what the physicians are using to kind of decide what type of chemo regimen should I go for. I mean, on the targeted therapy side, most popular one obviously is the HER2. There's also the PD-L1, but there are other things, like especially for prostate cancer, we have AR. TROP2 is a new drug. I'll actually talk about TROP2 and HER2 examples. And then there are several clinical trial biomarkers here as well. So why this emphasis on chemotherapy? This is actually a slide from gastric cancer space from Dr. Katnachi. If you really look into the advanced gastric cancer space, best standard of care where you're doing nothing, you're essentially seeing about 3 months overall survival. But you look at the different chemo regimens that you can go, whether you're using, all the E, for example, stands for epirubicin, doxorubicin, all these combinations. You can see actually there is a significant difference. This is a population-wide study in patients' outcome based on the regimen or chemotherapy regimen you give. Question becomes is, how do you choose what kind of chemotherapy? And in my knowledge, there's not many tests out there. How do you choose what kind of chemo regimen you should give? So this is where our reports go out and say, look, this patient is going to be is likely to be responding to epirubicin or doxorubicin, or has a resistance to a cisplatin or oxaliplatin based drug. So you might want, instead of using an EOF regimen, you might want to switch over to maybe an IF regimen and all that stuff. So that's chemo continues to be the backbone of most cancer types, actually. So that's one focus areas that we have. Over the years, we have run multiple types of cancer. Unfortunately, our prostate cancer database is very small because for some strange reason, we never ever focused on prostate. But we do have some prostate cancer samples in there. So this is just a graph showing you the HER2 protein distribution. We know the HER2 is very important in breast and gastric cancer, which is not unsurprising for us to see those levels. But we do see occasionally outliers of HER2 in different other cancer types. So here's an example of one cancer type like the head and neck cancer, in which the salivary gland carcinoma came in. And in the next few slides, I'm going to go really fast, but I'll just show you some case reports. So particularly, we had measured the levels of HER2, just as a point of reference. Anytime you're more than 750 attomoles per microgram, you are overexpressing HER2. So here's a patient from Johns Hopkins that got referred to us, and this patient had really high levels of HER2. They ended up treating the patient with a duo combination of 2 different anti-HER2 therapies, but given at the same time, Pertuzumab and Trastuzumab. And the patient, you can definitely see in the scans a vast improvement in their outcome. Something with we were chasing one particular patient in their cancer episode, where we are looking at targets for HER2, and the physician would go and give a Trastuzumab-based drug. But importantly, when you give a Trastuzumab-based drug, what happens is the cancer will find a way to evade that, and it will tend to upregulate the HER3 levels. So we saw that thing happening, and then he switched and gave a combination of Trastuzumab and Pertuzumab, and then immediately we are also seeing other mechanisms of resistance go up. So adding more and more drugs to this patient, and this patient actually had a pretty long survival compared to an advanced gastric cancer patient. I think it was 39 months compared to less than 12 months. Skip this, but this is more examples of where our work helped for some of these patients, especially in the gastric cancer space. That's with a drug that's in the market, right? Now, here's an example of a drug that's not in the market. It was in clinical trials at this point. We actually saw the levels of MET that was very high in this particular patient at 1480. The physician continued to go with the standard of care, and then with this chemotherapy, and then it actually bumped up because that seemed to be a favorite pathway, and that particular patient had 2090 the next time we examined. So he said, that's it. I'm going to put this patient into a phase 1 clinical trial. And then, you can see the scans from July to September, the particular patient got their cancer resolved. This is where we were able to help a physician make a decision on moving them into a clinical trial space. This paper is yet to come out. It's out online, but this is one thing that we most recently did in pancreatic cancer space. With the, as you know, pancreatic cancer is one of those horrible cancers. This particular patient, we had complete remission in HER2 pancreatic cancer. There's a whole slew of people in here, and you probably recognize some of these companies as well. And so,
well, one of them is XCures. So
yeah.
Emma might have been involved. I know that Emma's on the line.
Yeah, and
XCures Cancer Commons.
No,
yeah.
I'm sorry. I am not involved in XCures trials at all.
Okay. All right. I know there's a dichotomy there.
Yeah.
Yeah, Willie, who's
really, yeah,
I was with XCure, so
Yeah.
he's, yeah, and so Dan King, as he was at Stanford, and he was a fellow at Stanford when we did this work, but now he's moved on to Northwell, so he's continuing to work with us in the pancreatic cancer space. But this was one of those beautiful examples. Pancreatic cancers, they generally don't tend to have that much high HER2. I'm not going deep, but here you can see the mass spectrometry. We did not just the surgical specimen, 1870, remember 750 is your cutoff, right? 1870, 2070, 2018, look at that, it just bumped up from 1870 to almost 6,000. All right, this is the time when they said, time to put this patient on an anti-HER2 therapy. And if you know pancreatic cancer, it's usually less than a year. This is, at this point, it was 180 weeks out. And yeah, she's still doing very well. This is again, this is a case report of glioblastoma where a physician said, "I'm going to use your proteomics report and going to put this patient on irinotecan, which is literally, I mean, a cheap drug compared to everything else." Patient had recurrent glioblastoma and a PFS of 12 months. So this is now this report, and then something else that we are putting out in the brain cancer world, essentially that's showing that 15% of all glioblastomas tend to have very high levels of topo 1. Treat them with irinotecan. If you ask a normal quote unquote oncologist or a neuro-oncologist, they will say the use of irinotecan is kind of not, I don't want to do it because it has so much toxicity issues, people are vomiting and all that stuff. Agreed. But did you really look into whether your particular patient had high levels of topo 1? Then you might want to include a patient to give an irinotecan based therapy. More work on just some R&D work, but we've done a lot more work in a lot of different cancer types. Unfortunately, we still haven't done anything in prostate cancer. So most of the work's been focused on gastric and colorectal, understanding the mechanisms of resistance, either and several case reports and all that stuff. So let me switch gears a little bit here, and I need to talk to you all about this thing, because this is kind of becoming important in the space of HER2. So as you know, immunohistochemistry, we have 0s, 1s, 2s, and 3s. We got 277 breast cancer patient samples. This is work that we did with the Waldebron and MSK. As you can see, IHC will basically bin you into 4 different buckets based on the, I guess, the brown of the samples of the stain. But it was not unsurprising that the HER2 levels in IHC plus and overexpressor is variable. It's but we didn't really expect this the fold difference between the highest and the lowest one. And then what was kind of surprising for us, it was the IHC 0s and 1s which were historically not treated, or even some of the stews that were not amplified, they actually, like 50% or 25% of them in some instances, actually had some levels of HER2 in them. If you had asked this question about 2 to 3 years, people would have said, yeah, forget it, let's ignore the zeros and ones and twos, because that doesn't really have enough HER2 levels in them to be actionable. So we had this paper out, or this poster out in 14, and then paper came out in 15, 16 timeframe. But this particular paper, we examined some very high expressing HER2 levels and see how the overall survival was. So again, 740 or 750 is the cutoff for high overexpressing HER2. So long story short, this is those patients who had 2,200 nanomoles per microgram, or in other words, very high expressors, had no events at all. We're really looking at 6-7 years out when they were treated with an anti-HER2 therapy. Great. So this was kind of a first proof that mass spectrometry from FFPE tissue can predict the overall survival in these patients. So this paper essentially opened up the doors for us to do a lot more investigations with many institutions, MSK, University of Chicago, you name it. But going back to this particular figure here. So remember this IHC 3+ at least in the breast cancer space. This is roughly 15 to 20% if you step back and look at all the breast cancer folks. They're roughly 15 to 20%. The rest 80% is really the zeros, ones, and twos. And let's say the zeros are triple negative for this particular argument. And they are usually 15%. So the vast majority of the folks are really lying in this ones and twos. So fast forward a few years, there is a drug by the name Trastuzumab Deruxtecan, or Enhertu, that is from Daiichi AstraZeneca, and wherein they enroll the 1+ and the 2+ patients, this particular drug, and compared it to the chemotherapy arm. So the overall survival in this particular group of HER2 low, so we are classifying this as HER2 low, is 24 months compared to 17 months if you were just going with chemotherapy. This pretty much has opened the floodgates for the HER2 low population, and FDA recently approved this drug. Now, why is this important, and especially in the context of prostate cancer? I went back and said, hey, we had these 71 samples in our stock. One of the cohort said, let's examine what the HER2 levels in prostate cancer are. Something like 40% of them have no HER2, or we cannot detect HER2. About 5 patients or 3 patients or so have high levels of HER2. These were the patients that you would want to enroll into an anti-HER2 clinical trial. And hopefully they would do good. But with the low HER2 space opening up in breast cancer, this is a population that could potentially get this new drug. And in fact, there is a clinical trial that is going on that in prostate cancer, that is a combination of this particular drug, which is Enhertu or Trastuzumab Deruxtecan, and think this is a PARP inhibitor. And this is something I would encourage folks to actually look to know what your HER2 protein levels are. What and where in this chart, the prostate cancer would fall, right? And if it is high enough, there's a lot more options. If it is low enough, there's at least one option that's opened up. Let me switch gears a little bit here. I don't know if anybody has talked about this drug or antibody-drug conjugate. So the antibody-drug conjugate essentially is an antibody that is linked to a toxin with a linker. So that's just the basic format of the drug. So the antibody happens to go and bind to, let's say, HER2 in this case, and then it gets internalized, and then the toxin gets released, and based on the what toxin you use. If this toxin is towards tubulin inhibitors, you would go and bind to tubulin. The newer drugs are having the topo-1 inhibitor, so they go and bind to topoisomerase 1 and essentially prevent the cell division here. And some of these drugs also have a bystander activity. In other words, they can quote unquote get out of the cell and also kill the neighboring cells. So this is the more recent drugs tend to have the bystander activity as well. So the format is an antibody with a toxin.
Sheeno, just a comment about that. ADCs have definitely come up in my treatment recommendations. I know that I express very highly for B7H3, and so there's actually an ADC that targets B7H3.
No, perfect. And
I don't know if B7H3 is kind of like come up in your research, but there's actually a BiTE now from Xencor, which is in development, which targets B7H3.
So it is a BiTE, right, or an ADC?
There's two. So Daiichi has they have an ADC that targets B7H3, and Xencor is developing a BiTE that targets B7H3.
And it's a bispecific, right? So one is binding to the B7H3, and the other arm, I'm assuming this case is probably binding to a T cell and essentially getting the immune system to come and recognize that.
Correct. That is the Xencor product. Yes.
And if it is Daiichi, your B7H3 is your receptor, and your payload is actually going to go after topo-1. That's the format. The Deruxtecan is the payload, and that one goes after topo-1. So kind of knowing a topo-1 levels is good, but it's not mandatory, but it's definitely good. So we just kind of you set the stage up, because this is, and we were examining forget it again, ignore the cancer types, but this was just an adenoid cystic carcinoma and a type of head and neck cancer, very rare form. And if you look at, there are three possible ways if you had an ADC to AXL, and which at one point somebody had it. Either the receptor is not present, in which case forget it, the ADC cannot bind it, right? In the second scenario, the ADC can bind to it gets internalized, but once it is inside the cell, it has tubulin beta-3, which is a resistance mechanism for many anti-tubulin inhibitors. So that's not one that will be that effective. And then obviously the third scenario is, it works, it binds to it, gets inside the cell, and there's no resistance mechanism, and it should work. So this is kind of the three ways an ADC could work. I mean, it could be successful or not. Sorry, this is an extremely busy slide. Busy presentation. This was presented at AACR. There is a new thing out there called TROP2. The newest one, I should say, TROP2, and this is a receptor that is there on the cell. It is conjugated, or the Daiichi drug, sorry, not just the Daiichi, actually, Gilead's Immunomedics drug is a TROP2 that is conjugated to a topo-1 inhibitor. And Daiichi and AstraZeneca has a drug that is in clinical trials that is also doing the same thing, TROP2 conjugated to a topo-1 payload, okay? So we are examining something like 2,000 samples across different cancer types. And if you look at prostate, so the way you want to read this thing is 91% of prostate actually express TROP2 levels. So as you can see, not all of them express the same thing, right? There is anywhere from either very low levels to very high levels. So the minimum to the maximum was a 37x difference. So where in this group would somebody's prostate cancer be? The higher you are, the better. We did some work with MSK and looking at a median expression of TROP2 in triple negative breast cancer. We saw that in around 3,000 nanomoles. And the reason I'm talking about triple negative breast is it's approved, or the Gilead's drug is actually approved in triple negative breast. and it just definitely it was a TROP2 to topo1 combination. And I was kind of surprised, to be honest with you, how much prostate cancer samples had TROP2, and the fold difference is definitely 37x. Now, once it is internalized, let's look into what impact it is into a topo1 levels. So the prostate cancer, just chasing that figure, 92% of prostate cancers actually have topo1 with a very, I want to say, more tighter regulation of topoisomerase 1 levels. So this is a drug I'm actually excited to see how it will work in the prostate cancer space. And obviously, just looking at clinicaltrials.gov, there is 2 clinical trials that's ongoing. So IMMU is basically immunomedics that got bought out by Gilead. So it's now in the hands of the Gilead, but it's definitely in patients with metastatic castrate-resistant prostate cancer. I would again encourage you to at least know your TROP2 levels if you're high enough. And if it's an open label clinical trials, consider enrolling in one of these 2 trials. And Dato is the Daiichi AstraZeneca's drug, the same thing, it's TROP2 and topo1.
Sheeno, just a quick question on that. So are we just looking at RNA expression for TROP2? Is that what's going to tell us? Is that what's going to tell us whether or not we're a good candidate for these types of?
So in this particular case, that's a great question. So here's the thing. Let me escape this mode. These clinical trials, if you really look at them, they are not asking for any kind of expression. They're just saying, if you have prostate cancer, come on in.
Wow.
So my recommendation would be, especially if it is an open label clinical trial, right? Get yourself, figure out what the TROP2 protein levels are, and high enough, go and enroll in one of the open label, clinical trial. And that way you're stacking the odds in your favor.
Yeah.
And it could be you're a better candidate for TROP2, or maybe you're a better candidate for an anti-HER2 ADC clinical trial, right? Now, definitely ADC clinical trials are making waves. And so figuring out which clinical trial to enroll would be something handy here.
So Sheeno, I'm just going to drill on this just a second. So, I know my RNA expression, and there's some other patients on this call that may know it as well. And I know it relative to a few different cancer cohorts. And that's been really helpful because, in fact, it actually identified that I'm a very high expressor for B7H3 relative to even prostate cancer patients. But when you get into mass spec, are you saying that there can be differences in terms of the results that might come from RNA expression versus protein expression?
So let's put it this way. Not all RNA will express protein.
Okay.
Right? So for example, in some foundations, what they're doing is they're sending us samples so that to actually see if you're really expressing that protein or not. I mean, I know this is my extra slides, but there's one slide I have always kind of liked it. This is from Gordon Mills back in the day in 2015. And when you go from DNA to RNA to protein, how much loss of information really happens. This kind of has been replicated in many CPTAC and TCGA cohorts. So that is why, especially when it comes to cancer vaccines, or it's coming to in some trials, some of the patients are actually sending us the samples to say, do I express this protein or not? That's an additional comfort for them.
Yeah, this is super helpful. I think I've seen a variation on this. But so just so I understand, so you're saying there is a 35% delta between RNA and protein expression?
Oh, you mean from this slide? You mean?
Yeah, from that side,
So
or is it
what it is,
15%?
yeah, it's like if you go from DNA to RNA, right? Let's say 50% of your information,
Yeah.
only 50% information is transmitted, and then from RNA to protein,
Ah,
there is 30% of information that is translated and that gets down there.
yeah.
There's definitely a variation. I mean, this is again a long time ago.
Yep.
There are more and more data that come out. Definitely, the crux of the whole thing is like when you go from DNA all the way to protein, we know there is loss of information, and this is a recipe to make it, and this is the final product, if you will, right? And
Yeah.
all your drugs are actually acting against protein. So, and question then really becomes as should I measure the levels of protein to see if it's actually expressed, whether it be through mass spectrometry or whether it be through immunohistochemistry, whatnot, at least to know if the proteins are being expressed. That would be super helpful.
Got it. Thank you.
So I know it's 12:45, so I'm going to kind of stop here. Just want to let you know, all this work that we did is not coming from us, just us. We happy to collaborate with some really smart people from different parts of the world, and some of the smart people are listed here and from different parts of the world. So again, just that is a very small cohort of prostate cancer. I just wanted to examine at least a few targets and see what the distribution is. And I was kind of surprised to see the distribution, especially for TROP2 and HER2. We can go back at some point and actually start examining the 72 biomarkers in there and see what else could we find out within the prostate cancer space that not all patients are unique. That's kind of so for every patient, what works? What are the options if something's not working?
Great. So is that the end?
Yeah, that's it. Oh yeah, so here, last one. This was all extra slides, but the last one is basically, here's my contact information. If you plan on ordering any of these tests or your oncologist plans on ordering it, there's more information on our website, or you can send an email to orders@mprove.com. There's a quote unquote, I want to say a different website that we are migrating information from there to our main website. There's oncomicsdx.com that has a lot of information and papers in that as well.
Fantastic. This is really helpful. Thank you, Sheeno. And I have a number of questions, but I've already asked a couple along the way. And so I want to give other folks a chance to ask some questions. You can raise your hand or you can just jump in. Make sure you're off mute.
Assayed, how are you?
Assayed.
Yeah. Oh, very exciting presentation, actually. I really like the ADC part. And just about those type of drugs, how about the tissue specificity of those drugs? Say when you attach to a probes, that probes can connect to many different places, right?
Exactly. Especially
the receptor on the.
Oh yeah, absolutely. Especially one of the risk for the HER2 is the one that is most well understood. If you just take HER2 alone, outside of being just an ADC, just in a regular anti-HER2 therapy, it does have a cardiovascular risk, right? And now, with this particular ADC, where it was conjugated with topo1, they saw in 10% of the cases, which is actually now dropping down. Actually, they've learned better, and now it's maybe 1 or 2%. There is an interstitial lung disease, or pneumonitis, that is there with this anti-HER2 therapy, the Trastuzumab Deruxtecan. So it is a black box warning on the Enhertu drug, but the amount of survival difference that you're seeing in the breast cancer and even in gastric cancer is just staggering for this ADC. So it's definitely a risk-benefit analysis, but yeah, there's definitely a risk with it.
Okay, when you decide about the biomarkers, is it what type of the omics analysis to do? Is it the proteomic analysis? How do you detect your biomarkers, or that's just predefined
Yeah.
set of biomarkers?
So what we are really looking for the protein levels, right? So we are looking at the protein levels using mass spectrometry, and it is in our clinical lab, it is a predefined set of biomarkers. It is a 72 biomarkers we are looking for.
Okay.
And we'll swap out based on at some point, we'll change the composition of the biomarkers at some point, especially with newer drugs coming in space. The biomarker
Is it when you want to measure them? Are you measuring directly the protein, or are you measuring microRNA, mRNA?
No, we are measuring the protein, or I should say, we are measuring the peptide, which is a surrogate readout for the protein.
Okay.
Yeah.
Thank you.
Thanks, Assayed. Other questions? Okay, well, while others may be formulating some, Sheeno, I'm gonna jump in here. So you talked about using FFPE. Can you also use fresh frozen tissue?
We have in the past, but our clinical workflow is FFPE.
Okay.
So in some instances, when folks ask us, we'll send them the protocol to fix it. It's just essentially throw it into formalin and it fixes it, and then from there we go. Yeah.
Okay. I want to also comment on the speed at which you process. 5 days is really fast, particularly for mass spec. Just to give you like a benchmark, I'm getting whole exome sequencing from Tempus, and
Yes.
I'm looking at a month turnaround. So 5 days for mass spec is pretty quick.
Yeah, and the chances of failures are kind of high depending on the tissue type, right? In pancreatic cancer, we have seen that it's especially I'm not going to name names, but there are companies that will say, hey, it has failed because we didn't have enough material. So one thing recently that we have worked with another company, another genomics company, is we actually will try to do a laser microdissection of only the tumor areas and send it to the genomics company and highly enriched tumor. So now it works. And so that's rescued a few of the cases with the genomics testing.
Yeah, I've heard a little bit about that recently. That there's definitely some variation in WES, and we won't get into companies right now, but I'm aware of what you're referencing. You talked a little bit about the 72 biomarkers, and it sounds like you can customize the biomarkers that potentially we would want to target.
So our clinical workflow, we will keep it the same. It's 72 biomarkers, whatever is there right now. In our research panel, we honestly have something like 377 assays. So it's a research panel. So we have had some instances where folks will come, say, I would like to see Claudin 18.2. Is it expressed or not expressed? Or I don't know if, I think even B7H3 might be, actually reshare the screen. See, this is kind of going to be very small. B7H3 is part of our, I need to go back here and show you. There it is.
So I'm sorry. So your menu is 377, but you offer 72 of the 377. Sorry, that's a basic question. Okay, great.
That's right. That's the menu, right? 30 plus 42 is what we 72 is what we offer. But we have a menu of 377. This is again designed for R&D, designed for pharma. Sometimes they'll say, hey, we would like to pick and choose, and let's see what we can do. But this is mostly for R&D folks or pharma. In some instances, some of the patients of oncology will say, I want to see, do I express Claudin 18.2 or some of that's in here.
Yeah. So as I kind of think about this, so I've had my DNA sequenced a few times. I'm getting whole exome right now, so I'm going to get my RNA transcription data. Is it fair to say that I can use that as a guide to then determine which of these 377 we would want to focus on? I'm looking at the interplay between whole exome sequencing and mass spec and the proteins that you target.
And I would, because so here's the thing, right? With the RNA sequencing and stuff, you can actually see your 20,000 genes. You can see a much bigger picture, a bigger lens. Whereas with protein or proteomics, especially targeted proteomics, the lens is much smaller, or the menu is much smaller. So using that to guide whether I express this particular protein or not is a smart strategy.
Got it. Okay.
Because that's where the disconnect is, right? Did my RNA ever produce protein or not? So yeah.
No, that was super helpful. And that's kind of the direction that I was thinking of going. So this is super helpful. Richard Anders. Richard, I think you're new. Maybe if you can introduce yourself.
Sure. I'm an investor in early stage life science. I've been talking to Brad for a while. I have had cancer, but not prostate cancer, but I'm just interested in the hackathon model and ways that people can help themselves. So,
well, welcome.
Thank you. I just had a quick question about, what research you've done and what you can say about the heterogeneity or homogeneity of samples. Do you ever have people who sort of try to test multiple points within the sample? Because, if part of the sample that is free from a marker will probably lead to overexpression of that part of the sample. I just wonder what data you have on that subject.
That's a great question. This is a favorite question, but in general, what we say is the way we do laser microdissection, we try to get as many spots as possible and kind of average that heterogeneity out. If you're looking at, we call it the elevator experiment and the onion experiment, right? So where we are peeling off different layers of the onion, and we can see that when you're moving in a transverse plane, you really do see some heterogeneity. But when you're going down a tissue, at least for one section after the next section and the next section, we have put out papers. Actually, there's not that much difference that we tend to see, at least in the biomarkers that we examine.
So if there are multiple tumor regions, is it a good idea to sample them? Those might be transverse, but that's a really interesting point. But if there are multiple pinpoint regions, do you recommend, or is there any data on the advisability of sampling different regions?
So when the experiments that we did, again, we were looking at this was many years ago, and we were looking at a very small biomarker set. For those biomarker sets, we actually did not see that much difference. And the way we were trying to avoid that heterogeneity problem is we would sample, let's say, if we get a section of tissue and there is tumor spread out there, we would take out one pocket of tumor, and then we'll go out and go get another pocket of tumor, combine all of them, because this cancer type is going to be representative of, or the sampling would be representative of the cancer that you have, right? So that we can say, this is the biomarkers we saw in this particular panel. Yes, definitely that tumor heterogeneity problem is there. Make no mistake about it, it's definitely there. But the way we try to avoid it is we just try to get an average of the tumor. What the other experiment that I was describing is when you slice one section to the next section, if you look at the same area, which is slide 1, slide 2, slide 3, it's actually pretty even. I mean, you don't see much difference, at least until you go to maybe the 20th or 30th section, but until then, you don't really see much difference.
Yeah, no, that was a very interesting observation. I heard you say that one.
Yeah, that's we call it the elevator experiment. You went down, and other ones peeling the layers of the onion. Yeah,
and it makes sense. But yeah, it's really interesting.
We're at the top of the hour. I'm going to stop the recording, but everyone is more than welcome to stay on.